But I wanted to thank the organizers for the invitation to talk about multiple myeloma, especially considering the incredible expertise of the current moderators in the myeloma area. And there are multiple sessions on multiple myeloma tomorrow where you have additional great speakers. So what I tried to do here is just provide you with an overview of some of the clinical advances in myeloma biology and maybe a little bit about laboratory biology as well. And here are my disclosures. We know some things about myeloma biology, although certainly not as much as we would like. The development of myeloma is in part rooted in the normal development of plasma cells in response to antigenic stimuli. And unfortunately, at times, things can go wrong, and you can get, usually we think of the immunoglobulin heavy chain translocations or hyperdiploidy as early events, and then later on you get other events occurring, which can be things like deletion of 13, amplification of 1q, and then there are later things that can occur, and typically you have MGUS followed by smoldering myeloma, newly diagnosed, and then eventually relapsed and refractory disease. Again, thinking about the molecular level, this is the current dogma, which probably means it's about half right and half wrong. The problem is we don't know which half is which. But in general, the feeling is that, again, early events are IGH translocations or hyperdiploidy. Then you have some events that occur sort of in the middle of the disease, and then others which occur later in the relapsed refractory setting. But we know, for example, that dysregulation of p53 can occur through a number of mechanisms, not just deletion of 17p, and the same thing with MYC. The classical MYC translocations seen in Burkitt, for example, are rare, but there are other ways that you can enhance MYC expression. And then this map just gives you an overall feeling of how complex myeloma really is, especially in the newly diagnosed setting. You would think that maybe there's going to be only a couple of changes, but in fact there are many copy number gains and losses as well as translocations. And this in part has made it a fascinating disease to study in the laboratory, but a little bit more frustrating from a clinical perspective because there is not one or two or three abnormalities in all patients that you can target. And as you can imagine, when you get to the relapsed and refractory setting, this map becomes even more complicated. So I wanted just to briefly start with the current diagnostic criteria for MGUS and AMN. MGUS, or monoclonal gammopathy of undetermined significance, essentially are patients who have a usually low-level monoclonal protein, usually a low level of bone marrow involvement, and don't have any disease-related symptoms, which are things like hypercalcemia, renal insufficiency, anemia, bone lesions, or certain molecular markers. And then asymptomatic or smoldering myeloma are essentially the same, but have a higher M protein, at least 10% marrow plasma cells, and no disease-related symptoms. I think when I went to medical school, we all thought that MGUS, and that was a long time ago, by the way, but it's still true, we all thought that MGUS was relatively benign, and you followed these people and nothing much happened. But in fact, we know now that although certainly MGUS is nowhere near as bad as myeloma, there are some problems that these patients face. And here from a recent paper from Bob Kyle, who's the godfather of myeloma and MGUS in particular, you can see that compared to the expected outcome of patients that are age and sex matched, the observed outcome for patients with MGUS is not as good, and they do develop higher risks of other cancers. So there is definitely some kind of additional dysfunction here, and we should not just necessarily say, oh good, you know, your MGUS hasn't changed in the past year, don't see me again for 5 years. These are definitely people who need more stringent follow-up. Looking at the smoldering category, for many years, this was a very frustrating disease for patients as well as physicians Because typically, I would imagine as a patient, the last thing that you want to hear is, oh, you have cancer. And then the next worst thing to hear is, oh, and by the way, we're not really going to do anything about it. We're just going to watch and wait. I think that's still probably the standard of care, but the Spanish did a study some years ago now showing that lenalidomide and dexamethasone compared to observation had a longer progression-free survival for the treatment group and also a longer overall survival. And recently, this paper, by the way, should be appearing relatively soon in the Journal of Clinical Oncology, but this presentation at ASCO showed the results of this ECOG trial where patients were randomized either to observation or to just lenalidomide, so no dexamethasone here. And here are the mature progression-free survival data where you can see that compared to observation, you have a relatively low level of progression with lenalidomide, with a hazard ratio that I think we would all be very happy if the trials that we led met. Here, if you like the numbers, you can see a low rate of progression on len versus a much higher rate of progression on observation. People who are purists will point out that the overall survival so far has not been different in this study, and that's true. So I think there will be a debate. Should we treat these people if they don't live longer, or is it just that the data have not yet sufficiently matured? But we may be now changing the paradigm in smoldering, certainly on the intermediate and high risk, to maybe treat early rather than just watch and wait. A little bit about what we're doing at MD Anderson, and Elizabeth Manasank at our center is the leader of these studies. We have an observational cohort where we're following several hundred patients with MGUS or smoldering myeloma, and in addition to molecular studies, which others have done before, in fairness, and not seen dramatic differences at progression, but we're also doing immunologic profiling on a regular basis to try to understand whether there is immune escape mechanisms that contribute to progression from MGUS to smoldering, and then smoldering to active myeloma. And along the way, we've also done some interventional trials, including one study that just appeared in Blood Advances in the pilot anti-PD-1 antibody phase, and another of anti-CD38. Here's just a little bit of the early data about the observational study. You can see that, for example, here, gene expression profiling of patients who have progression does show that not only are their myeloma cells different, but also their microenvironmental cells are different. So clearly there is some interaction here. And these were data collected when we had a relatively low number of progressors. We now have many more progressors, which is not good for patients, but good in terms of collecting data. So we're hoping to be able to contribute something to the immunologic understanding of maybe triggers that result in progression. We also completed a study of pembrolizumab. The rationale here was in part data from a SWOG study and our own trial showing that higher levels of PD-1, PD-L1, and PD-L2 predicted for more rapid progression. And this study, unfortunately, was stopped because of the concerns that later emerged from Merck trials of pembrolizumab with immunomodulatory drugs. But we did enroll enough patients to at least show a proof of concept. And we had one lady who achieved a complete remission. This was a lady after just 3 doses of pembro who had 40% plasma cells at baseline with deletion of 17p. Probably otherwise would have absolutely progressed within a year, definitely 2, and yet now is 4 years out in molecular complete remission. So unfortunately, we probably won't be doing pembro in this setting for other patients, but it does show that immune therapy can have some promise here. And then we also just completed a study of isatuximab, which is an anti-CD38 monoclonal antibody similar to daratumumab. and that showed a 50 to 60% response rate, which is pretty good for a single agent. Unfortunately, the FDA set the bar at 70%, so this will probably not be moving forward as a single agent, but there are plans for combination studies with lenalidomide, and Dr. Gabriel, for example, is going to be leading one of these international studies, which will look at ISA with Len-Dex as the experimental arm. We also currently are looking at a personalized neoantigen vaccine approach, and although right now we're doing this in smoldering myeloma, it may also be something to consider in the future for patients with MGUS, considering the low risk and toxicity of this approach. But in our observational study, we found that it was fairly easy to detect and predict neoantigens. So essentially, we're taking bone marrow from these folks, we're analyzing their DNA and RNA, we're using predictive algorithms to try to determine what antigens will best be presented, and then they get a cocktail of 10 peptides representing different mutations. We've enrolled the first 10 patients so far. There's been no toxicity, and we're waiting for the immune correlates. But these are hopefully things that will be of use and value in the future. I wanted also to talk a little bit about patients with symptomatic myeloma, and these are the current NCCN guidelines for transplant-eligible patients, many of which have been determined in part by incredibly nice studies done by the IFM here in France, and bortezomib, len, and dexamethasone, and bortezomib cyclophosphamide and dexamethasone, as well as bortezomib with thalidomide and dexamethasone, are probably worldwide accounting for 90% of the upfront therapies. One of the ways this will probably be changing in the future is through the addition of monoclonal antibodies. There are several such studies, and in the interest of time, I only pulled out one of them, which is the so-called Griffin trial that's been led by Peter Voorhees in the US, where essentially the control arm was bortezomib, len-dex, transplant, and then len maintenance, whereas the experimental arm added daratumumab, the anti-CD38 antibody, and this did show a higher stringent complete remission rate, going from 32% to 42%, and also seemed to induce higher levels of MRD, or minimal residual disease negativity. And hopefully this study will be published in Blood relatively quickly, but I think it does show you that we're going to be doing combination chemo-immunotherapy as the front line for most patients with myeloma in the future. Transplant is still a relatively wonderful standard of care after induction, but we do know that auto-transplant is not curative. One thing that we're doing at MD Anderson is building on a natural killer cell platform, and we've been able to show, particularly in high-risk patients, these are small pilot studies, so we have to be a little bit careful, but NK cells do have anti-myeloma activity, and you can see compared with patients getting a standard of care autologous transplant, we've been able to show that just with NK cells alone, you can, it looks like, induce better quality responses and more MRD negativity. Most recently, we've added elotuzumab. The rationale is in part that these stimulate the NK cells and also help to target myeloma. It looks like MRD negativity is even higher. And then we're also considering changing these NK cells, which right now are not particularly targeted, but by turning them into CAR NK cells. Maintenance is also a standard of care after transplant. To me, maintenance is actually a failure, because if you could eliminate myeloma 100%, you wouldn't need to continue other drugs afterwards. But since we haven't gotten there yet, we do maintenance. Len is probably the standard of care. And one thing we've done is to look at combinations like elotuzumab, which is the other currently approved monoclonal antibody in myeloma, which targets a protein called SLAMF7, And although this is a non-randomized study, it does show a 2-year estimated progression-free survival of the combination that looks better than would be the case for len alone. And we are now doing a study through IACH and SWOG and the alliance in the US of len versus len-dara in maintenance. And then after 2 years, if you're MRD negative, you're going to be randomized further either to stop maintenance or to continue maintenance. So it'll be interesting to see what happens there. For transplant ineligible patients, which typically are folks that are a little bit older and more frail, you have some similar combinations as options, but one of the recent developments here, also led by Thierry Facon here, was the addition of daratumumab. So sort of a mirror image of the transplant-eligible patients trying to improve therapy by adding immunotherapies. And this was a large randomized study looking at len-dex versus dara-len-dex. And you may probably know that the hazard ratio for this was again quite good and showed a better progression-free survival for the combination of dara, len, and dex. In the relapsed or refractory setting, and there will be special talks about this tomorrow, so I just kind of threw up the current NCCN guidelines. And the good news is that there are lots of options, as you can see, many of which are category 1, which means that they are based on randomized phase 3 trials. The bad news is that there are lots of options, and rarely do you see the different options compared head-to-head with each other. So it's tough to know what the best sequence is and what the best therapies are for individual patients. But just briefly touching on some of the new drugs that are out there, you earlier on this session heard about selinexor. The first approval for selinexor turns out to be in myeloma, where you can see that there were patients that had good responses, although also quite a few that did not, and median progression-free survival was 2 to 3 months, while overall survival was 9 months. And although these numbers are not as robust as we would like, you do have to remember most of these patients were what are called penta-refractory, which means disease that had progressed after prior bortezomib, carfilzomib, lenalidomide, pomalidomide, and daratumumab. And in combinations, for example, with bortezomib or carfilzomib, we expect that these numbers will be even better. So definitely a welcome addition to what we can do for myeloma. Probably our first molecularly targeted therapy will be venetoclax, which is a BCL2 inhibitor. It turns out that patients with the 11-14 translocation, which is about 15% of folks, so it is the most common translocation that we see, have high levels of BCL2. So they have very high overall response rates. This is with carfilzomib and dexamethasone, but really any regimen containing venetoclax should be given to patients with 11-14 in the relapsed setting, and one could see maybe even incorporating that as a frontline for 11-14 patients in the future. Getting back to the immunotherapy theme, a number of BiTEs, or so-called bispecific T-cell engagers, are out there in clinical trials. These are essentially similar to monoclonal antibodies, but instead of there being 2 FAB fragments that go after the same target, usually one arm will go after a T-cell, and a second arm will go after a protein on the surface of your favorite cancer cell, and the T-cell becomes activated and attacks. So AMG 420, which is the first BiTE that we have reasonable data for in myeloma, which goes after BCMA, or B-cell maturation antigen, which is one of the best, I think, targets for myeloma because it's relatively plasma cell specific. You can see a 70% response rate in relapsed and refractory disease, including some people that achieved complete remission with MRD negativity, although the stringency of the MRD testing was not as high as perhaps some of us would have liked. but it does give you a rapid response and a pretty good median time of MRD negative CR duration. Other ways to target BCMA include with antibody drug conjugates. This will probably be the first BCMA-targeted therapy to be approved in the U.S. This is an antibody called belantamab mafodotin. You can see a 60% overall response rate. Again, many of these patients with quad and penta-refractory disease, essentially the antibody binds BCMA, it's internalized, and it releases the conjugated drug, which is an antimicrotubule agent. A little bit of laboratory data from our group that was an oral presentation at last year's ASH. There are other BCMA antibody drug conjugates, and here's one that we've worked with preclinically, which will be in the clinic probably next year, early maybe January, February. But this is a BCMA antibody conjugated to amanitin, and you can see actually in mice a single dose of this drug is enough to get you a complete remission, and we followed these mice out to 200 days. Usually if they don't relapse within 100 days, it's considered a quote-unquote cure, and even 200 or more days they're cured. So we would love if the same thing hopefully happens in patients as well. And then the last way to target BCMA is with CAR T-cells, and there are again sessions tomorrow about this, so I won't go into it in a lot of detail, because there are actually many CAR T-cell studies. But essentially this is another way to target T-cells towards an antigen of your preference. And from one of Dr. Gobriel's colleagues in the Boston area, Nupur Raj, earlier this year were the first larger data sets published with a product called BB2121. And you can see that if you get a reasonable dose of the CAR T-cells, you have a good progression-free survival of about 1 year. And if you're in CR and MRD negative, which is almost half of patients, these numbers get even better. So clearly, a lot of promise here. We hope to soon see patients being cured, especially as these therapies are moved earlier, but definitely in the relapse refractory setting, something to look forward to. So just some conclusions in terms of initial therapy. I think early intervention for high-risk MGUS and smoldering myeloma is in our future. I think it's a promising area to go after. That's a little bit of a pun because Dr. Gobriel is leading a trial called PROMISE in the United States, which is trying also to identify predictors of progression from MGUS to smoldering myeloma. For transplant-eligible patients, some kind of triplet with an immunotherapy is probably going to be the standard of care. And for transplant-ineligible patients, probably a doublet with immunotherapy, just because I think trying to get 4 drugs into these older patients may be a little bit difficult. Stem cell transplant is still a standard of care, but CAR T cells may wind up being better. I think the question is how much better, and will the cost of these, which will be more than an auto transplant, be worth the incremental benefit in terms of outcome. In the relapsed and refractory setting, second-line progression, we have many wonderful therapies, which include proteasome inhibitors, immunomodulatory drugs, and antibodies. In later lines, we have venetoclax for 11-14 translocated patients. In more advanced disease, we now have agents like selinexor and BiTEs and ADCs and CAR T cells, although we still need to do lots of clinical trials to define this better. If we have time, there also could be a discussion about what the future of myeloma therapy could look like. And in case we don't have time, Dr. Kanachua and I published this overview in Annual Review of Medicine earlier this year, where we divided people up into those who were fit and frail, and then into molecularly low or high risk, and looked at doing, for example, for low risk patients, pretty much things that we're doing now, which would include 2 or 3 drug induction regimens. followed probably by stem cell transplant and then maintenance, whereas for high risk, maybe 4 drug induction regimens, maybe CAR T cells as a consolidation, and some kind of combination for maintenance, whereas if you're frail and low risk, you could do 2 or 3 drug regimens, maybe an antibody drug conjugate as a consolidation, because these would not be eligible for transplant, whereas over here you might maybe do a BiTE instead of CAR T cells as a consolidation, and then either single agent or combination maintenance. And also in terms of relapse disease and various refractory disease options, fortunately there are possibilities in all of these areas, but none of these are yet curative, so there's a lot that we can do. Thanks very much for your attention, and happy to answer any questions.